Hello everyone, welcome to the next video of our this series that is basically focusing to discuss all the key concepts behind enzyme or how does enzyme works and all the things. In last video we have basically discussed the basic introductory part of enzyme and our today's agenda is to discuss the main concept, how does enzyme works. In a simple layman terms, which we also discussed in our last class, that the basic function which enzyme actually does, that is catalyzing any biochemical reaction. Catalyzing any biochemical reaction. Now, the next point comes in your mind. Okay, fine, I understand enzyme what enzyme does, but how? How does enzyme able to achieve this much of efficiency in this? For this, discuss something about the reaction thermodynamics. First thing which you'll understand, reaction thermodynamics. Let's draw the reaction curves. That is this, this. It can be like, we can label it like that progress of reaction. And in y-axis, we can label it like free energy. We all know that whenever any reactions takes place, it's because of its stability. Because the free energy of the finished products should be lesser than the free energy of any substrate, because lesser the free energy, more the stability. Thus the reaction thermodynamics curve is basically it's like this only. In this curve you can understand there's basically 3 points. One point is like this, one point is like this, and another point is this. Now what does these points means? This is the highest point, and this point is called transition point. And what happened in this transition point? The basically in this transition point, the reactant transit into the product formation. The product formation begins at the transition point only. Means this is the highest point of, and this is the amount of energy, and this is also called activation point. Reaction activation point. The energy gap in between this, the substrate form and the transition point, this is called activation energy. And this difference, the free energy difference between the product and the substrate, this is called free energy change of the reaction or delta G reaction. I guess things are not clear. Why any reaction takes place and how we can identify which reaction will take place and which will not take place. Now, basically, what enzyme does, enzyme basically affects this activation energy. It doesn't have any effect on this. In last class, he also mentioned one thing. Enzyme does not affect reaction equilibrium. Thus, enzyme have no effect on the delta G value of any reaction. Enzyme basically reduces this activation energy. Now, how? In this graph, we can easily understand the thing. When enzyme binds to this substrate, is basically from enzyme substrate complex, and that is bit stable. And this is from enzyme substrate complex. Then again, substrate transfer into products. This is from a enzyme product complex, and from these products will be like this and enzyme release. This is the basic thing we can understand. This is enzyme substrate. This is enzyme product. Now, from this what we have understand? In normal state, the free energy is like this much, but in this special case, the free energy is only like this much. Means this amount of free energy actually reduces, and by this way, enzyme catalyzing the reaction. Now get into a bit deeper into these as well. If- Now suppose this is an enzyme. This is an enzyme. Yes, I'm not good at that much drawing. Just please consider. And this is the substrate. Now you can easily understand this is the point where basically these substrates come and bind. What is this bind is called? This is called active site. Means the substrate binding site present on the enzyme is called active site. When this substrate binds to this active site, there is some liberation of some energies as well. This is called binding energy. And this binding energy somewhere this compensate this activation energy. I guess up to this we all are in the same page. Now this hypothesis. Now let's talk about some hypothesis. How this binding takes place. There is basically two hypotheses. One is called lock and key model. In this model, basically the scientists say like the enzyme binding sites as well as the substrate both are rigid in nature. That's why as the if any key fits to any lock, then the key can actually open this lock, that lock. If an enzyme and a substrate perfectly matches its configuration or its structure, then the substrate can bind to the enzyme and only then this reaction catalyzing can takes place. And another point, another hypothesis that is induced fit model. Induced fit model. In this model, what they have suggested that this active site is not rigid. Active site is flexible. Based on the compatibility in between the enzyme and the substrate, the active site can take the shape of this, the complementary shape of the substrate, and by this way, it both can bind and further reactions can take place. Means the key difference between lock and key model, you can lock and key model along with the induced fit model. In lock and key model, active site is rigid, but in the induced fit model, active site is not rigid. This is flexible. Now, let's get into deeper, more deeper in this. We can understand this substrate. This is the active site. Both take some binds, and then by this, there's some release some binding energy, somehow compensating the activation energy, and all the things we are clear. But when this active site, this substrate is binding to this active site, there should have some bonding, and there should have some strategy by which they actually catalyze. Now let's talk about catalyzing strategy. There are basically three things again. What are the bonds is possible in between Uh, substrate and enzyme, which is first point is covalent bond, means the bonds are very strong, and this is observed in which kind of which enzyme, that is chymotrypsin. Second standard should be acid base reaction. In a simple term, we all know what is acid base reaction. That is the electron given or proton transfer reaction. This acid base reaction, what is the most prominent example for acid base reaction? That is lysozyme. And in our last video also, we have observed one thing. Metal is an important factor. Based on the presence of metal as a cofactor, we actually identify classifying enzyme as metal activated enzyme, metal enzyme, and metal is an important part. And these metal ions are actually classified in another strategy, that is metal ion catalysis. And as a metal, this is considered within Lewis acid base reaction. I guess till time we all in the same page. We already understand what are the sites of enzyme, where the substrate can bind, and the further reactions takes place. What could be the possible things in between the in wasn't responsible for this attraction between this enzyme and substrate? It could be the structure, it could be the separate bonds, and because of this bond, there should have some release of energy. Apart from this energy somehow uh, influencing the normal reaction thermodynamics, and it is decreasing the activation energy of that reaction, and by this way enzyme reaction catalysis takes place. Now whenever we're talking about something function, there should have some parameters, means by this we can identify this much works that an enzyme does. Thus the enzyme activity should have some units, and actually it has. Let's talk about enzyme activity units. Now, when we draw the units, there should have some, there's a two CGS, most dominating one, CGS and SI. The CGS unit is enzyme unit, or in short form is called U, and SI unit is called kata, or it is called KAT. This enzyme unit is basically is equals to, sorry, 1 micromole minute inverse. What does it mean? It basically signifies amount of enzyme requires to convert 1 micromole amount of substrate into product in a span of 1 minute of time. I guess this is now this is clear. This is time duration, and this is amount of substrate. Now what is the value of catal? That is 1 mole second inverse. Now, similar of enzyme units, we can just how we can define this one catal. Amount of enzyme requires to convert 1 mole of substrate into product in the span of only 1 second. I guess all the things are now clear. If we just visit this much, that should have some bonus, because this is a bonus season. This question is important for gate actually. Not there should have some relation between one enzyme unit and one catalyst as well, right? So we will define that relation now. Now, what is one enzyme unit? 1 U equals to 1 micromole by 1 minute. Now 1 micromole is equals to what? 10 to the minus 6 mole, and 1 minute is equals to 60 seconds. Now calculate this. The value is 16.67 into 10 to the minus 9. This is mole per second. Now equals to, we can write 16.67. 10 to the power 9 means nano. Mole per second means cattle. Thus, what we can understand from this? 1 enzyme units equals to 16.67 nanocat. And these type of questions are actually important for gate as well. Okay, I think now you all are in the same page, and we can all actually understand how does enzyme works, and how the function signifies, and what is the basic key things, factors present within those interactions which are actually responsible for this catalyzing. This is all for now. In the next class, we will get delve deeper into the enzyme kinetics part as well. Thank you.